Title: Assessing the Probability of Alien Visitation to Earth. Science, Speculation, and Evidence. The question of whether extraterrestrial beings have visited Earth is both captivating and controversial, straddling the boundaries of science, philosophy, and cultural mythology. Scientifically, the probability of alien life existing elsewhere in the universe is widely regarded as high due to the vastness of space, the estimated hundreds of billions of galaxies, each containing billions of stars and potentially habitable planets. The Drake equation, proposed by Frank Drake in 1961, attempts to estimate the number of technologically advanced civilizations in the Milky Way galaxy alone, factoring in star formation rates, the fraction of stars with planets, and the likelihood of life developing and becoming intelligent. While this equation offers a framework, its variables are speculative, leading to a broad range of possible outcomes, from none to millions of civilizations. However, asserting that such civilizations are not only real but have also traveled vast interstellar distances to visit Earth is a far more contentious claim. The primary scientific objection to the alien visitation hypothesis is the immense distance between stars, with even our closest neighbor, Proxima Centauri, being over 4 light-years away. At current technological capabilities, such a journey would take tens of thousands of years. This constraint necessitates either a civilization with propulsion technologies vastly beyond our understanding or the use of concepts like wormholes or warp drives, which remain speculative and unproven within the framework of general relativity. Additionally, there is a conspicuous lack of empirical evidence. While UFO, now UAP, or unidentified anomalous phenomena, sightings abound, no credible, peer-reviewed data have confirmed extraterrestrial origin. The U.S. government's recent reports on UAPs acknowledge that some phenomena remain unexplained, but they do not constitute proof of alien visitation. Psychologists and sociologists also note the role of cognitive biases, hoaxes, and cultural influences in perpetuating alien visitation beliefs. Nevertheless, the field of astrobiology continues to expand our understanding of life's potential. Discoveries of extremophiles, organisms that thrive in extreme Earth environments, suggest that life could exist in diverse planetary conditions, and missions like the James Webb Space Telescope aim to analyze exoplanet atmospheres for biosignatures. The scientific community generally embraces the search for extraterrestrial life but remains skeptical of direct contact narratives without rigorous proof. In essence, while the probability of life elsewhere in the universe may be high, the probability that such life has visited Earth remains extraordinarily low based on current evidence and scientific understanding. Title: Alien Life and the Drake Equation. Science, Speculation, and Belief. The Drake equation, proposed by astronomer Frank Drake in 1961, is a probabilistic framework used to estimate the number of technologically advanced civilizations in our galaxy. It incorporates variables such as the rate of star formation, the fraction of stars with planetary systems, the number of habitable planets per system, the probability of life developing, the emergence of intelligence, the lifespan of technological civilizations, and their ability to communicate across interstellar distances. While scientifically useful for framing questions about extraterrestrial life, the equation's reliability is limited by the fact that many of its parameters remain speculative or based on single data points, Earth being our only confirmed case of life. Thus, the Drake equation is more a conceptual tool than a predictive model, useful for guiding astrobiological thought but not yet offering definitive answers. As of now, no scientific mission has definitively found evidence of alien life, though several have come close to identifying promising environments. Missions like NASA's Perseverance rover on Mars are currently analyzing surface samples for ancient microbial life, and the James Webb Space Telescope, JWST, is scanning exoplanet atmospheres for biosignatures, chemical indicators like methane and oxygen that could suggest biological processes. Additionally, studies of Enceladus and Europa, moons of Saturn and Jupiter respectively, have revealed subsurface oceans with the potential for extremophile life. While none of these missions have yielded direct proof, they significantly expand the scope of environments considered potentially habitable, making the search increasingly data-driven and multidisciplinary. Despite the absence of concrete evidence, belief in alien visitations remains widespread, largely due to psychological, cultural, and perceptual factors. Cognitive biases like pattern recognition and confirmation bias make humans inclined to interpret ambiguous stimuli, lights in the sky, shadows in photos, as meaningful. Cultural saturation through movies, documentaries, and social media reinforces these interpretations. Additionally, the idea of alien visitors fulfills a deep narrative craving, a cosmic mystery that both challenges and reassures. Reports of unidentified aerial phenomena, UAPs, especially those acknowledged in government briefings, further blur the lines between skepticism and belief, despite lacking empirical grounding. In this way, alien visitation beliefs persist not from evidence, but from a compelling mix of curiosity, mythology, and modern myth-making. Title: The Hunt for Alien Life, Promising Worlds, Webb's Impact, and UAP Controversy. Among the most promising planets for discovering alien life are exoplanets located in the habitable zones, the Goldilocks region where conditions might allow liquid water to exist. Top contenders include TRAPPIST-1e, LHS 1140b, and K2-18b, which not only orbit within their stars' habitable zones but also show potential for Earth-like atmospheres. K2-18b, in particular, has drawn attention after spectral data from the James Webb Space Telescope, JWST, suggested the presence of carbon-bearing molecules like methane and carbon dioxide, possibly linked to biological activity. Closer to home, Mars remains a key candidate for ancient microbial life, while Europa and Enceladus, Jupiter and Saturn's moons, harbor subsurface oceans and have shown signs of hydrothermal activity, raising hopes for extremophile-like ecosystems. The James Webb Space Telescope is transforming SETI, Search for Extraterrestrial Intelligence, and Astrobiology by providing the first high-resolution spectroscopic data on exoplanet atmospheres. Its ability to detect biosignatures, such as oxygen, methane, or dimethyl sulfide, can indicate biological processes, especially when these gases appear in unlikely chemical combinations. JWST is also capable of identifying surface temperatures, water vapor, and cloud coverage, offering a multidimensional picture of planetary environments. While JWST is not designed to detect radio signals like traditional SETI tools, it significantly broadens the search for life by enabling the characterization of potentially habitable worlds, turning the theoretical parameters of the Drake equation into testable science. As for UAPs, unidentified aerial phenomena, their scientific credibility remains limited despite recent governmental disclosures. The 2021 and 2022 U.S. intelligence reports acknowledged that some sightings remain unexplained but emphasized that they lack evidence of extraterrestrial origin. Most UAPs likely stem from misidentified aircraft, drones, atmospheric phenomena, or sensor artifacts. The limited data, lack of physical evidence, and secrecy surrounding many reports make them difficult to analyze scientifically. While UAP research is becoming more rigorous, particularly with NASA's 2023 UAP Independent Study, it currently resides more in the realm of speculative investigation than confirmed science. Title: Tracing Alien Life, Methane Mysteries, Ocean Worlds, and JWST's Next-Gen Vision. Methane detection on exoplanets is highly significant because it can serve as a potential biosignature gas, a chemical indicator of life. On Earth, the vast majority of methane is biologically produced by microbes called methanogens, although geological processes like volcanic activity or serpentinization can also generate it abiotically. What makes methane particularly intriguing is when it appears alongside gases like oxygen or carbon dioxide, forming a disequilibrium that is difficult to maintain without a continuous source, typically biological. For example, the detection of methane on K2-18b by the James Webb Space Telescope, JWST, especially when combined with carbon dioxide and possible dimethyl sulfide, suggests conditions that could support or indicate life. However, scientists must carefully rule out abiotic processes before attributing methane to living organisms, especially on planets with vastly different conditions than Earth. The icy moons Europa, Jupiter, and Enceladus, Saturn, are among the most promising locations in our solar system for microbial life due to their subsurface oceans, which lie beneath thick ice crusts. These oceans are kept warm by tidal heating, gravitational interactions with their parent planets, which may also drive hydrothermal activity on the ocean floors, analogous to Earth's deep-sea vents where life thrives without sunlight. Enceladus has been observed ejecting plumes of water vapor containing organic molecules, suggesting the presence of a dynamic, potentially habitable environment. Europa Clipper, launching in 2024, and JWST, ESA's Jupiter Icy Moons Explorer, aim to further explore these environments, searching for signs of habitability or even direct biosignatures. JWST differs significantly from the Hubble Space Telescope in its ability to search for life. While Hubble revolutionized our understanding of the cosmos through visible and ultraviolet light, JWST operates primarily in the infrared spectrum, which is crucial for analyzing the chemical compositions of exoplanet atmospheres. JWST's advanced spectrometers can detect minute changes in starlight as it filters through a planet's atmosphere during transits, revealing the presence of gases like water vapor, methane, carbon dioxide, and oxygen. Unlike Hubble, JWST can characterize temperatures, cloud layers, and pressure profiles, key elements for assessing habitability. In this way, JWST transforms exoplanet science from detection to detailed atmospheric analysis, inching us closer to identifying potential life beyond Earth. Title: Life Signs in the Stars. Methane, Ocean Worlds, and JWST's Astrobiological Revolution. Methane alone cannot definitively prove the existence of life on another planet, but it is considered a promising biosignature gas when found under the right conditions. While a significant portion of Earth's methane is biologically produced, abiotic processes such as serpentinization, rock-water interactions, photochemistry, and volcanism can also generate methane. Therefore, its detection is only meaningful when it appears in chemical disequilibrium, especially alongside oxidizing gases like oxygen or carbon dioxide, which tend to react with methane and remove it from the atmosphere. If methane is present in unexpectedly high concentrations and is continuously replenished, it raises compelling questions, but still requires contextual data, such as surface conditions, atmospheric dynamics, and the absence of known non-biological sources, to support a case for life. Multiple upcoming and current missions are targeting Europa and Enceladus, two of the most promising worlds for discovering extraterrestrial life within our solar system. Europa Clipper, launching in 2024 by NASA, will perform nearly 50 flybys of Europa to map its surface, measure its ice shell thickness, and assess the composition of its potential subsurface ocean. It will also search for organic molecules and thermal activity near the Moon's surface. The European Space Agency's JWST, Jupiter Icy Moons Explorer, launched in 2023, will explore Europa, Ganymede, and Callisto, focusing on subsurface oceans and habitability. As for Enceladus, though no dedicated mission is confirmed yet, NASA's proposed Enceladus Orbalander concept would orbit and then land to analyze plume ejecta and subsurface chemistry directly, potentially sampling materials from the ocean without drilling through ice. The James Webb Space Telescope, JWST, is poised to reshape the future of astrobiology by transforming the search for alien life from speculation into quantifiable chemical investigation. By using infrared spectroscopy, JWST can analyze the atmospheres of Earth-sized exoplanets in the habitable zones of nearby stars, detecting potential biosignatures such as methane, ozone, water vapor, and carbon dioxide. Unlike previous telescopes that focused on detecting planets, JWST enables atmospheric characterization, temperature profiling, and even assessments of planetary weather and cloud cover. These capabilities allow scientists to assess whether planets might support life-sustaining environments or exhibit atmospheric compositions inconsistent with abiotic chemistry alone. In doing so, JWST moves the field of astrobiology into a data-rich era where potential life detection becomes measurable rather than merely hypothetical. Title: Probing Alien Oceans, Sampling Europa's Plumes, Methane-Oxygen Synergy, and JWST's Life Target Worlds. Sampling Europa's plumes without landing is a key innovation in planetary science, offering a low-risk, high-reward strategy to investigate potential biosignatures. These plumes, believed to be jets of water vapor and ice particles expelled through cracks in the Moon's icy shell, can be intercepted by spacecraft during close flybys. Instruments such as mass spectrometers, dust analyzers, and organic molecule detectors can directly analyze the ejected material, capturing its chemical composition in real time. NASA's Europa Clipper will perform this technique, using tools like the SUDA, Surface Dust Analyzer, and MASSPEX, mass spectrometer for planetary exploration, to search for salts, organics, and potentially complex molecules that may originate from Europa's subsurface ocean, eliminating the need to penetrate its kilometers-thick ice shell. Methane becomes a much stronger biosignature when paired with oxygen or other oxidants like ozone or carbon dioxide in a planetary atmosphere. These gases tend to chemically react with methane, breaking it down relatively quickly. On Earth, their co-presence is maintained by biological replenishment, oxygen from photosynthesis and methane from microbial life. In exoplanetary atmospheres, finding both in abundant, stable concentrations suggests an ongoing process replenishing both gases, which is hard to explain through known abiotic means. This disequilibrium serves as a red flag for life, especially if it occurs on rocky planets in the habitable zone with Earth-like temperatures and liquid water. The James Webb Space Telescope, JWST, is actively targeting a selection of temperate, Earth-sized exoplanets in its first phase of atmospheric characterization. Notable candidates include TRAPPIST-1e and TRAPPIST-1f, part of a 7-planet system 40 light-years away, with several in the habitable zone. LHS 1140b, a super-Earth that orbits a quiet red dwarf, ideal for detailed spectroscopy. K2-18b, where JWST has already detected signs of carbon-bearing molecules and water vapor, suggesting a potentially habitable environment. GJ1214b, a mini-Neptune, being studied to understand thick atmospheres and water vapor retention. These planets are chosen based on their transit geometry, stellar brightness, and low stellar activity, which maximize JWST's ability to detect faint atmospheric signals. As its capabilities unfold, JWST will continue to shape the search for life with unprecedented spectral precision, focusing on biosignature-rich targets that may redefine our place in the universe. Title: Landing on Ocean Moons, Biosignature Chemistry, and JWST's Habitable Horizon. Future probes could indeed land on Europa or Enceladus to sample their subsurface oceans more directly, but such missions present formidable technical and planetary protection challenges. On Europa, the ice shell may be 15 to 25 kilometers thick, requiring either deep drilling or the use of cryobots, robotic landers equipped with melting probes, to reach the liquid water below. NASA's Europa Clipper, launching in 2024, will not land but may pave the way for a future lander, such as the conceptual Europa lander, which could analyze surface ice for biological molecules. Enceladus offers a more accessible opportunity, as its geyser-like plumes continuously eject subsurface material into space. A proposed Enceladus orbolander could sample these plumes from orbit and then land to study surface composition, enabling direct analysis of potentially life-bearing oceanic material without needing to breach the ice. The most compelling gas combinations for detecting life involve pairs or sets of gases that are thermodynamically unstable and require active replenishment, hallmarks of biological processes. The strongest example is methane, CH4, and oxygen, O2, coexisting in significant concentrations. On Earth, this balance is maintained by life. Plants and algae produce oxygen, while microbes generate methane. In an abiotic environment, These gases would rapidly react and neutralize each other. Other powerful combinations include O2 or ozone, O3, with nitrous oxide, N2O, methane with carbon dioxide, CO2, in the absence of carbon monoxide, CO, dimethyl sulfide, DMS, a molecule typically produced by marine plankton, if detected alone. These combinations, when detected in the atmosphere of a rocky planet in the habitable zone, could signal biological activity, especially when supported by planetary data indicating liquid water and moderate temperatures. As of early 2025, the James Webb Space Telescope, JWST, has begun atmospheric studies of over a dozen exoplanets, with at least 6 targets located in or near their stars' habitable zones. These include TRAPPIST-1e, F and G, K2-18b, LHS-1140b, and GJ-1214b. While full biosignature confirmation is still pending, JWST has already detected water vapor, carbon-bearing molecules, and possible disequilibrium gases in several atmospheres. The telescope's precision has set a new standard in exoplanetary science, enabling scientists to not only catalog planets but to assess their potential for hosting life based on actual atmospheric chemistry. JWST is expected to study dozens more exoplanets over the next decade, exponentially increasing our understanding of alien habitability. Title: Countdown to Discovery. Future Landers, Life's Chemical Signature, and JWST's Expanding Horizon. A lander mission to Europa could realistically arrive in the early to mid-2030s, depending on funding and mission development progress. NASA's Europa Clipper, launching in 2024, will conduct multiple flybys to scout Europa's surface and identify ideal landing zones. While the Clipper is not equipped to land, it sets the stage for the proposed Europa lander, which could follow later in the 2030s if prioritized by NASA and Congress. The lander would aim to analyze surface ice for complex organic molecules and potential biosignatures. As for Enceladus, NASA's Enceladus Orbolander, a high-priority concept recommended by the 2022 Decadal Survey, could launch in the late 2030s, with arrival in the 2040s, depending on funding. The Orbolander would both orbit Enceladus to sample plume ejecta and then land on its surface to analyze materials ejected from the subsurface ocean. The most unambiguous gas signature of life would be the detection of a strong redox disequilibrium, specifically the simultaneous presence of oxygen, O2, and methane, CH4, in an exoplanet's atmosphere, sustained over geological time. This combination indicates active replenishment since these gases rapidly react and eliminate each other. On Earth, this balance is a hallmark of biological activity. Other promising biosignatures include nitrous oxide, N2O, and dimethyl sulfide, DMS, both difficult to produce abiotically, and even potential unknown exotic biosignatures tailored to non-Earth-like life. However, methane and oxygen together, especially in an environment with liquid water and Earth-like temperatures, remain the gold standard for inferring life from afar. By 2030, the James Webb Space Telescope, JWST, is projected to have studied dozens of habitable zone exoplanets, possibly reaching 50 to 100 atmospheric analyses, depending on telescope time allocation and mission extensions. Current targets like TRAPPIST-1e, LHS-1140b, K2-18b, and others will be supplemented by new discoveries from ongoing missions like TESS, Transiting Exoplanet Survey Satellite, and the upcoming Plato, ESA, mission. JWST's ability to capture infrared spectra with extreme precision will allow it to characterize atmospheres, detect weather patterns, and identify potential biosignatures in a growing catalog of nearby rocky worlds. If any one of these planets shows the right chemical fingerprints, it could provide the first compelling evidence of life beyond Earth, before the decade ends. Title: Biosignature Limits, Planetary Protection, and the Long Voyage to Enceladus. The James Webb Space Telescope, JWST, is theoretically capable of detecting complex biosignature gases like dimethyl sulfide, DMS, and nitrous oxide, N2O, in exoplanet atmospheres, but only under specific and favorable conditions. Both gases are compelling because they are predominantly produced by biological processes on Earth, DMS by marine plankton and N2O by microbial activity in soils and oceans. However, their detectability depends on factors such as planet size, atmospheric density, distance from Earth, and the gas's concentration. For now, JWST's sensitivity is best suited for larger, close-in exoplanets orbiting quiet M-dwarf stars, where atmospheric signals are stronger. While not yet confirmed in any exoplanetary spectra, simulations show that high concentrations of DMS or N2O could be detectable if they exist in thick atmospheres on nearby planets like TRAPPIST-1e or K2-18b. The risks of biological contamination on Europa and Enceladus are significant concerns governed by strict planetary protection protocols. Both moons are considered Category 4 bodies by COSPER, Committee on Space Research, meaning they have a high potential for hosting life. If Earth microbes were to survive spacecraft sterilization and contaminate these worlds, they could either compete with or mimic native life, compromising our ability to identify genuine biosignatures. To mitigate this, landers and orbiters undergo extreme sterilization measures, including heat treatment, chemical cleaning, and cleanroom assembly, to meet bioburden limits. In missions like Europa Clipper or a future Enceladus Orbalander, the spacecraft will avoid impact with the surface at mission end, preventing accidental contamination in case of control loss. Reaching Saturn's moon Enceladus is a substantial interplanetary endeavor that takes approximately 6 to 13 years, depending on propulsion technology and planetary alignment. For example, Cassini, which explored Saturn and Enceladus from 2004 to 2017, took 6.7 years using gravity assists from Venus, Earth, and Jupiter. A future Enceladus mission, like the Enceladus Orbalander, could use similar slingshot maneuvers or opt for faster but fuel-intensive propulsion systems. The flight duration is influenced by mission objectives, orbit-only versus lander, payload mass, and launch energy. Efficient yet robust trajectory planning is essential for balancing time, cost, and scientific return, particularly when targeting such a distant and promising world. Title: Lightspeed Dreams and Molecular Clues, Future Propulsion, Biological Gases, and Safeguarding Alien Life Detection. Laser propulsion, a futuristic technology under active research, has the potential to dramatically shorten travel times to the outer solar system. Unlike chemical rockets, which carry heavy fuel, laser propulsion systems use ground-based or space-based lasers to push lightweight sails or mirrors attached to spacecraft, enabling constant acceleration. Projects like Breakthrough Starshot envision sending gram-scale probes to Alpha Centauri at 20% the speed of light using this method. For interplanetary missions, scaled-down laser sails could reduce travel time to Enceladus or Europa from a decade to just a few years, if payload constraints and power delivery can be solved. However, the technology is still experimental, facing challenges like beam targeting, sail stability, energy losses, and deceleration at destination. Still, it represents one of the most promising long-term solutions for rapid deep space travel. Dimethyl sulfide, DMS, and nitrous oxide, N2O, are compelling biosignatures because they are primarily produced by biological processes and have few plausible abiotic pathways in Earth-like environments. DMS is emitted almost exclusively by marine phytoplankton as a byproduct of sulfur metabolism. In planetary atmospheres, its presence at detectable levels would strongly imply a global biosphere, particularly if associated with water vapor and moderate temperatures. N2O is produced by denitrifying bacteria in soil and aquatic ecosystems, and while it can form abiotically under specific conditions, such instances are rare and require extremely reducing environments. Both gases would be especially significant if found in chemical disequilibrium with other oxidants, strengthening the case for life. To ensure life detection integrity on ocean worlds like Europa and Enceladus, scientists employ stringent planetary protection protocols. These include the sterilization of spacecraft components, often through dry heat microbial reduction, alcohol baths, or hydrogen peroxide vapor. All spacecraft are assembled in ISO Class 5 cleanrooms, and microbial samples are regularly taken to ensure bioburden remains below allowable thresholds. Missions must also plan for end-of-life disposal, such as controlled orbits or crash avoidance, to prevent accidental contamination. When detecting potential biosignatures, researchers apply strict false positive elimination criteria, comparing detected molecules against known abiotic sources. Any potential life evidence is subjected to multiple redundant instruments and cross-checking to rule out contamination, analytical artifacts, or abiotic analogs. This cautious, multi-layered approach ensures that if signs of life are found, they are as unambiguous and reproducible as possible. Title: Navigating Deep Space and Discerning Life. Laser Limits, Exotic Biosignatures, and Scientific Rigor. Laser-propelled interplanetary missions, while revolutionary in concept, face several technical and physical limitations that currently constrain their practical deployment. First, they require extremely high-powered laser arrays, often in the gigawatt range, to accelerate spacecraft, systems that do not yet exist at the necessary scale. Second, these missions demand ultralight sail materials capable of withstanding extreme heat and maintaining structural integrity under high acceleration. Precision beam focusing and alignment across astronomical distances also pose a challenge, especially to keep the beam trained on a tiny sail for days or weeks. Lastly, decelerating the probe upon arrival is a major hurdle. With no onboard fuel, alternative methods like magnetic or photonic braking must be developed. While promising for small, uncrewed probes, laser propulsion is not yet viable for larger, data-intensive missions like orbiters or landers needed for in-depth planetary study. Dimethyl sulfide, DMS, and nitrous oxide, N2O, could theoretically exist in non-Earth-like atmospheres, but their interpretation as biosignatures becomes more complex. In extremely reducing atmospheres, abiotic pathways for N2O production, such as lightning or high-energy UV-driven photochemistry, might occur, although still considered rare and inefficient compared to biological output. DMS, in contrast, has no known robust abiotic production mechanisms and remains more tightly linked to life. The presence of these gases in thick, hydrogen-rich exoplanet atmospheres, such as that of K2-18b, is still scientifically intriguing but demands context-specific analysis, considering the planet's pressure, temperature, UV flux, and chemical stability. Thus, while the gases remain compelling, their detection outside Earth-like contexts must be interpreted with caution. Avoiding false positives in detecting alien life requires a robust, multi-pronged scientific methodology. First, scientists use contextual planetary modeling to understand the full range of abiotic processes that could produce a given gas under that planet's conditions. Second, they rely on multiple biosignature gases appearing together, especially when in thermodynamic disequilibrium, such as methane and oxygen coexisting. Third, cross-validation through different instruments, spectrometers, mass analyzers, imaging systems, helps rule out detection artifacts. Fourth, Earth-based laboratory simulations replicate atmospheric chemistry to test which gases can arise naturally. Finally, scientists aim for repeatability, multiple observations across time and with different methods to confirm a consistent, unexplained pattern. Only when these rigorous standards are met can a potential biosignature claim move beyond speculation and toward probable evidence of life. Title: Advancing Light-Speed Travel and Biosignature Integrity. Next Steps in Space Exploration and Life Detection. The next milestone for laser sail propulsion lies in small-scale, proof-of-concept missions that validate core technologies such as beam steering, sail materials, and acceleration mechanics. Organizations like Breakthrough Starshot and university-led initiatives are developing centimeter-scale StarChip probes coupled with ultra-thin sails that could be launched into low Earth orbit or the Moon's vicinity to demonstrate photon propulsion under real conditions. These missions aim to test sail durability under laser bombardment, the effectiveness of attitude control, and telemetry during high-velocity acceleration. If successful, they will pave the way for interplanetary versions capable of reaching the outer planets in just a few years, serving as precursors to eventual interstellar missions. To determine whether dimethyl sulfide, DMS, can arise through non-biological means, scientists could engineer lab-based and planetary analog experiments simulating a wide range of atmospheric, geological, and photochemical conditions. This would include replicating hydrothermal vents, lightning discharge environments, and sulfur-rich planetary atmospheres without life, to test whether DMS can be synthesized abiotically at detectable levels. No such process has yet been verified on Earth or in simulations, making this research critical to validate DMS as a truly robust biosignature. Future planetary probes to Venus or Titan, where sulfur compounds are abundant, might also test for abiotic DMS production in non-Earth-like chemistries. While there is no fixed number, the presence of two or more biosignature gases in chemical disequilibrium is generally regarded as the minimum threshold for a confident life claim. The most compelling pairs include methane, CH4, and oxygen, O2, or ozone, O3. Nitrous oxide, N2O, with oxygen or CO2. DMS with oxygen or water vapor. These combinations indicate that multiple life processes are operating simultaneously, such as photosynthesis and methanogenesis, that are hard to replicate abiotically. The more diverse and incompatible the gas set, the stronger the case. Additionally, these gases must be observed over time and at concentrations inconsistent with geological or photochemical equilibrium, reinforcing the hypothesis of active biological maintenance. Ultimately, confidence arises not from a specific count, but from the collective improbability of such a chemical profile in a non-biological context. Title: Mapping the Final Frontier and Redefining Life Signals. StarChip, Biosignatures, and Atmospheric Context. StarChip, a proposed gram-scale spacecraft propelled by ground-based lasers, is primarily designed for interstellar exploration, targeting Alpha Centauri. However, scaled-down prototypes could feasibly be tested in the Solar System before 2035. If flight-tested in the 2030 to 2032 window, StarChip models could reach the outer planets in just 2 to 4 years, depending on acceleration success and trajectory. These flyby probes would not be able to map in traditional high-resolution terms, but could capture key magnetic, radiation, and dust data, along with low-resolution imagery of targets like Neptune, Uranus, or Kuiper Belt objects. Given StarChip's limited onboard power and instrument payload, it would function as a scouting platform, paving the way for more capable laser sail explorers. Biosignature standards in exoplanet science are rapidly evolving in response to new technologies like JWST and future missions like LUVOIR and HabEx. Early biosignature models focused on oxygen or water vapor alone, but now scientists emphasize biosignature suites, combinations of gases, climate models, and planetary conditions. The field is increasingly incorporating probabilistic frameworks, where the likelihood of life is assessed based on statistical inference, Bayesian analysis, and contextual planet data. This shift includes assessing false positives and false negatives, quantifying how likely abiotic processes could explain the signal, and prioritizing observable disequilibrium across multiple gas species. Researchers are also exploring non-Earth-centric biosignatures, including sulfur, nitrogen, and exotic chemistry, to account for life in non-terrestrial environments. Methane-oxygen disequilibrium is unlikely to persist in completely waterless atmospheres, as water plays a crucial role in the generation, maintenance, and destruction of both gases. On Earth, photosynthesis and microbial methanogenesis are water-dependent processes, and atmospheric water vapor influences photolytic reactions that govern methane breakdown. Without water, sustaining large quantities of both gases would be chemically unstable unless there is extremely low UV flux, strong shielding mechanisms, or alternative energy gradients maintaining separation. Thus, while not impossible, methane-oxygen disequilibrium in dry atmospheres is considered less reliable as a biosignature, and its presence in such a context would demand robust abiotic modeling to rule out geochemical oddities or planetary anomalies. Title: Beyond the Known Earths: Redirecting StarChip, Next-Gen Telescopes, and Life's Extremophile Signals. Redirecting StarChip prototypes to capture exoplanet data presents major challenges due to their high-speed flyby design and lack of onboard propulsion for navigation or braking. While feasible for flyby imaging of planets or stars they pass near, their rapid velocities, potentially 20% the speed of light, mean they would have mere seconds to minutes to capture data. To study exoplanets in detail, StarChip would need advanced miniaturized sensors, onboard AI, and laser relay systems to transmit meaningful data across light years. Given the constraints of power, bandwidth, and stabilization, these probes are best suited to scouting missions or one-time snapshots, not sustained observations. Future versions might include targeted missions to Oort cloud objects or rogue planets, but for detailed exoplanet analysis, traditional telescopic observatories remain superior. The upcoming space observatories LUVOIR, Large UV/Optical/IR Surveyor, and HabEx, Habitable Exoplanet Observatory, are designed to surpass JWST in the direct detection and imaging of Earth-like exoplanets. LUVOIR will offer a much larger mirror, 8 to 15 meters, and broader wavelength coverage, enabling the direct imaging of dozens to hundreds of habitable zone planets with high-resolution spectroscopy. Its powerful coronagraph and potential starshade will block starlight to reveal faint planetary signatures, allowing precise measurement of biosignature gases like O2, CH4, H2O, N2O, and even surface reflectivity, vegetation red edge. HabEx, though smaller, focuses on characterizing Earth-sized planets around Sun-like stars, with a dedicated architecture for biosignature confirmation. Both will offer longer baseline missions, greater sensitivity, and the ability to distinguish between life and false positives more confidently than JWST, marking a transformative leap for astrobiology. While most biosignature models center on Earth-like or oceanic worlds, researchers have begun exploring theoretical biosignatures for extreme environments like desert planets or magma worlds. For arid planets, possible signs include H2-dominated atmospheres with trace organics, or high levels of acetylene, ethylene, or ammonia, which could suggest hydrogen metabolizing life. In high-temperature environments, above 400 degrees Celsius, scientists have proposed that silicon-based or metal oxide life might emit novel gases like silane, CH4, or phosphine, pH3. While speculative, such biosignatures would depend on extremophile metabolic analogs and would require observatories like LUVOIR or future X-ray/UV telescopes to detect. These signatures must be interpreted in the context of geology, atmospheric dynamics, and thermal stability, making them harder to confirm but valuable targets for broadening the search beyond Earth-centric assumptions. Title: Silicon Life, Starshades, and the Limits of Discovery. Pushing astrobiology beyond the ordinary. Silicon-based life, while entirely hypothetical, could potentially produce detectable atmospheric gases, though none have been observed in nature. Silicon, like carbon, can form long chains and stable molecules, but it is less chemically flexible and forms less diverse compounds under Earth-like conditions. However, in high-temperature or reducing environments, theorists suggest that silicon-based organisms might metabolize or emit compounds such as silane, CH4, a volatile silicon analog to methane. Other candidates include silicon oxides or organosilicon compounds, depending on atmospheric chemistry. If life existed on lava worlds, tidally heated exoplanets, or metal-rich deserts, such emissions might be detectable with infrared or UV spectroscopy, especially if present in anomalously high concentrations inconsistent with geological sources. While no silicon-based biosignature has been confirmed, including such possibilities in biosignature modeling widens the search for non-carbon-centric life in extreme planetary environments. Starshades are external occulters, giant, flower-shaped spacecraft that fly tens of thousands of kilometers in front of telescopes to block out starlight, enabling direct imaging of faint exoplanets nearby. Unlike coronagraphs, which are internal and limited by telescope optics, starshades offer cleaner contrast and broader wavelength performance. In missions like HabEx or LUVOIR, starshades would allow telescopes to image Earth-sized planets around Sun-like stars with minimal starlight interference, making it possible to detect reflected light spectra that carry biosignature gases. Starshades are especially useful for observing multi-planet systems, improving planet detection efficiency, and reducing observation time per target, dramatically enhancing the science return of next-generation observatories. LUVOIR, with its large aperture and ultraviolet 2-infrared coverage, could feasibly detect signs of life on rogue planets or tidally locked exoplanets, provided the right atmospheric conditions. While rogue planets, which float freely without a host star, are dark and cold, some may retain heat through internal radioactive decay or thick insulating atmospheres, supporting subsurface or greenhouse-warmed biospheres. Tidally locked planets, common around M-dwarfs, have one side perpetually facing their star. LUVOIR can observe the terminator zones, the habitable twilight belts between day and night sides, using high-resolution spectroscopy to detect water vapor, methane, or temperature gradients consistent with surface liquid water. These marginal environments might host microbial life and offer new frontiers for astrobiology beyond conventional Earth analogs. Title: Aligning Vision and Life, Starshade Precision, Silicon Metabolism, and the Habitability of Twilight Worlds. Starshades coordinate with space telescopes through a highly choreographed process involving precise formation flying, advanced telemetry, and predictive orbital modeling. Typically positioned 30,000 to 100,000 kilometers away from the observing telescope, like LUVOIR or HabEx, the starshade must align within 1 meter of accuracy across tens of thousands of kilometers to perfectly block the host star's light while allowing exoplanet light to pass unobstructed. This is achieved using a combination of laser beacons, optical sensors, autonomous navigation, and ground-based commands. After aligning with one star system, the starshade must reposition itself, a process that can take several days to weeks due to the vast distances and fuel-efficient trajectories required. Though technically demanding, this setup dramatically improves imaging contrast, making it possible to detect Earth-sized planets orbiting Sun-like stars, especially in multi-planet systems. On Earth, while no known microbes metabolize silicon as a primary energy source, some bacteria interact with silicon in biologically relevant ways. Certain diatoms and radiolarians incorporate silica, CO2, into their cell walls, using silicon transport proteins. Additionally, silicon-utilizing pathways exist in bacteria involved in biomineralization, and hyperthermophiles can survive in high silica environments such as hydrothermal vents. While these do not indicate true silicon metabolism, akin to carbon or sulfur pathways, they suggest that life can evolve to interact with and structure itself around silicon, especially in hot, high-pressure environments. This hints that in non-Earth-like conditions, such as those on lava worlds or rocky exoplanets, silicon-based metabolic analogs might emerge, particularly if carbon is scarce. Terminator zones, the twilight belts between day and night on tidally locked exoplanets, are considered prime candidates for potential habitability because they represent thermal, atmospheric, and chemical stability zones. These planets have one side perpetually facing their star, day side, and the other in permanent darkness, night side. But along the terminator, temperatures may be moderate enough to allow liquid water. Models suggest that atmospheric circulation and ocean heat transport could prevent atmospheric collapse on the night side and excessive heating on the day side, creating a stable climate region. Furthermore, sunlight at shallow angles could enable photosynthesis-like processes without the harsh radiation exposure of full daylight. For exoplanets around M-dwarfs, where tidal locking is common, terminator zones represent one of the most likely locations for life to persist outside Earth-like rotational environments. Title: Life at the Edge of Light, Habitability of Terminator Zones, Starshade Maneuvers, and the Limits of Silica-Based Biology. Terminator zones on tidally locked exoplanets could plausibly support complex, multicellular life, especially if conditions remain stable over geological timescales. These zones provide moderate temperatures, a steady if oblique supply of stellar light, and the potential for robust atmospheric and oceanic circulation that distributes heat and nutrients across the planet. Climate models suggest that such regions could develop temperate microclimates with liquid water, avoiding the extremes of both day and night sides. If photosynthetic processes can adapt to low-angle light and if atmospheric chemistry supports biosynthesis, multicellular organisms, possibly adapted to consistent twilight or varying temperatures, could evolve. The presence of magnetic fields, plate tectonics, and a stable atmosphere would further enhance habitability, making terminator zones promising candidates not just for microbial life, but for ecological complexity. Starshades must reposition every time a new star system is targeted, which could range from once every few days to a few weeks, depending on the mission plan and target list. Because starshades operate at distances of tens of thousands of kilometers from their partnered telescopes, even slight movements require significant time, fuel, and precision. Efficient repositioning strategies involve gravity assists, low-thrust ion engines, or solar sail maneuvers to conserve propellant while maintaining alignment accuracy. As a result, missions may plan for dozens of starshade repositionings over several years, balancing time between imaging sessions and transit periods. Careful sequencing maximizes science return by targeting clusters of nearby stars or pre-identified exoplanet-rich systems. Silica-based life forms, though hypothetical, are unlikely to be viable under Martian or Venusian conditions due to chemical and physical constraints. On Mars, the cold, dry, and oxidizing environment would challenge the stability of silicon-based macromolecules, which are generally less versatile and more brittle than carbon analogues. Venus, though rich in atmospheric sulfur and CO2, features temperatures above 460 degrees Celsius and extreme pressure, which might allow some exotic chemistry but would degrade known silicon compounds unless encapsulated in highly resilient matrices. However, some theorists propose that silicon-based membranes or skeletal frameworks could exist in extremely reducing or high-pressure environments, such as those on hot exoplanets or deep inside planetary crusts. While no known Earth organisms metabolize silica, extremophiles like hyperthermophilic archaea interact with silicate surfaces, hinting at possible prebiotic roles for silicon in alien biochemistries, but not yet a full biological scaffold for life. Title: Sustaining Life in the Shadows, Twilight Energy, Silicon Skeletons, and the Mechanics of Starshade Missions. In twilight, terminator zones on tidally locked planets, life could draw energy from a mix of sunlight, geothermal activity, and atmospheric gradients. Although sunlight arrives at shallow angles and with lower intensity, it may still be sufficient for photosynthesis, particularly if adapted pigments evolve to capture broader or red-shifted spectra. Analogous to shade-adapted plants or deep-sea photosynthesizers on Earth, organisms might thrive in these dim environments. Additionally, if the planet has active plate tectonics or tidal heating, especially near M-dwarfs, geothermal vents could provide localized heat and chemical nutrients, supporting chemosynthetic life. Temperature and wind differentials between day and night sides may also generate atmospheric currents usable by aerobic or anaerobic microbial metabolisms, enhancing energy access in ecosystems that bridge the day-night divide. Silicon-based skeletons could theoretically support soft-bodied life in high-gravity environments, where structural reinforcement is essential to resist compression. Silicon, particularly in the form of silica, CO2, or silicon carbide, forms rigid, high-strength frameworks that could serve as exoskeletal or endoskeletal scaffolds. On Earth, diatoms and sponges already use silica for support, albeit in aquatic settings. In high-gravity worlds, such structural adaptations would reduce bodily deformation and enhance support for internal organs. However, the metabolic cost of synthesizing and maintaining silica frameworks, especially in dry or low-energy conditions, might limit their use to slower-growing, mineral-recycling organisms or to forms that rely on bio-mineral absorption from the environment rather than synthesis. These life forms could resemble crystal-armored invertebrates, thriving in mineral-rich, high-pressure zones. A 5-year Starshade mission could sustain 30 to 50 target maneuvers, depending on mission architecture, propulsion type, and starfield density. Each maneuver, where the Starshade realigns with the telescope to image a new star system, may take from a few days to over 2 weeks, including travel and stabilization time. The number is constrained by available propellant, solar illumination for power, and instrument calibration time post-realignment. Efficient sequencing, such as observing multiple exoplanets in one stellar neighborhood, can reduce repositioning demands and maximize scientific yield. Advanced starshades may eventually use ion propulsion or solar sails to increase fuel efficiency, potentially expanding the number of repositionings per mission and enabling a broad exoplanet census across diverse systems. Title: Beyond Carbon, Mineral Life, Starshade Precision, and the Geologic Wellsprings of Life. While life on Earth is built around organic carbon, theoretical frameworks have proposed that mineral-based life, organisms structured and possibly energized through inorganic substrates, might be possible, though highly exotic. Such life would not use carbon polymers like proteins or DNA but could be organized through crystalline scaffolds, metal oxides, or silicates, possibly using electron transfer networks instead of carbon-based metabolism. On Earth, life forms such as electrogenic bacteria, e.g., Geobacter and Shewanella, already use iron and manganese oxides to transfer electrons for energy, hinting at the feasibility of metabolism linked to minerals. In environments where organic chemistry is rare, like magma worlds, lava tubes, or deep planetary crusts, a purely mineral-based biochemistry might operate on longer evolutionary timescales, with slower metabolisms, perhaps even as silicon crystal lattice organisms. However, no such life has been observed, and the energy efficiency, error correction, and information storage systems of non-carbon frameworks remain speculative. To manage drift and misalignment, starshades rely on precision formation flying, using star trackers, gyroscopes, accelerometers, and optical beacons to align with their paired telescope from tens of thousands of kilometers away. Small thrusters, often ion engines or cold gas propulsion systems, perform fine-tuned adjustments to maintain the line-of-sight blockage of a target star. Autonomous control algorithms continuously monitor position and orientation, comparing onboard sensor data with predicted orbital paths. Any drift is corrected using pre-programmed feedback loops or ground-based updates. The Starshade structure also includes flexible petal-shaped edges, carefully engineered to diffract light in a way that minimizes residual glare, making sub-meter alignment accuracy achievable even in deep space. Geothermal biospheres are supported by geological conditions where heat, fluid circulation, and chemical energy converge. These include mid-ocean ridges and hydrothermal vents, where tectonic activity allows seawater to percolate into hot crust, dissolving minerals and returning as chemically rich plumes. Subduction zones, where one tectonic plate dives beneath another, generating superheated fluids and gas emissions that sustain microbial communities. Volcanic calderas and lava tubes, which trap heat and allow for isolated ecosystems. Serpentinization zones, where ultramafic rocks interact with water to produce hydrogen and methane, vital for chemosynthetic life. Ice-covered oceans on moons like Europa and Enceladus, where tidal heating drives oceanic convection and vent-like chemistry. These geologic features create redox gradients, heat flux, and mineral scaffolds, providing both energy and structure for life, often independent of sunlight. On other planets, similar conditions could form the basis of subsurface or crustal biospheres, broadening the scope of life's possible abodes.